Hello everyone! Who is ready to deep dive into this article right here? Because I know I am. Platelet hyaluronidase 2 regulates the early stages of inflammatory disease and colitis. This was actually a very fundamental paper for my thesis, so I'm very excited to talk about this with you all. Also, say hi to my old boss and my mentor. Hi, Aaron! What is this paper about exactly? Well, here's the brief description.
Paper summary as told by Science Goblin. When sick, long sugar grow on cell. Very sticky. When no longer sick, platelet go slice. Boop. But with IBD, platelet no slice. Why platelet no slice? Platelet lost sword.
Yep, I have no dignity left. But yep, that's pretty much it. That's the paper. Hot dang, this was a fast video. Gonna make me a celebratory cup of tea and, oh, you're still here? Oh, you can see the video length. Oh yeah, there's more to come. Let's go. Paper background as told by a boring lady with a PhD. We're going to be discussing in this paper something called IBD, which stands for inflammatory bowel disease. What this will look like is patchy inflammation all along your gut. And there's two types that you need to know about. It's Crohn's disease, which is where you have patchy inflammation. Ulcerative colitis is more localized in your lower intestine. It doesn't go all the way along your intestinal tract. But you will be seeing both of these throughout the paper. Now, there's a couple of things that are known in IBD. We know that in your gut, we have endothelial cells here, here, here. So this is it's not your gut lining. This is actually your veins. This is carrying the blood that will go into your gut. What is known is that we have immune cells that when you have a flare-up during IBD, they will go into your tissue from the bloodstream and they will start to wreak havoc. We call this an increase in inflammation. So when you have inappropriate recruitment of immune cells, this is going to cause your immune system to go haywire. And so that's where you're getting these patches from, or you're getting these just intense pain areas in your gut. It's because there's been recruitment. So what is the thing that causes this inappropriate recruitment? And there are a lot of reasons, but the one that this lab focuses on is a long molecule called hyaluronan. So we have hyaluronan, or HA. You'll see both. Right. So what I have drawn it, though, is in its anti-inflammatory form. What does that mean? Well, what that means is that this form here, where it looks like a long, long barrier, blocks the recruitment of immune cells. It keeps the immune cells from going in. So that's why it's called anti-inflammatory. However, when you are sick, you will start to build these long chains of HA into the environment, or extracellular environment, I should say. It is important to note that what makes up these heavy chain HAs is something called I-alpha I. This will be important for some of the experiments that we'll see in the paper. But for now, you just need to know that these are heavy chain HA. A, or HCHA. Now, these are called pro-inflammatory. Why? Well, because they actually mediate and encourage immune cell infiltration. So, when you're not sick, you have the running parallel, non-inflammatory HA, and when you are sick, that's where you get these long chains. Okay, but here's the thing. That's how your body's supposed to work. What's supposed to then happen is that, let's zoom in on those HA cables. You're actually supposed to start cleaving them because, well, you don't need them anymore when you're not sick. And so that's where you're going to start to get HA fragmentation. This is HA metabolism. And this is what happens when you're healthy. However, when you're sick, let's erase these really quick. However, during IBD, you actually lose this HA metabolism. So you continue to have these long, heavy chain HA that will continue to recruit immune cells. This is less than ideal for a lot of reasons. So the lab was trying to figure out, why are we losing this HA metabolism? What could it possibly be? There actually is an enzyme that we're going to look at, and it's called HYAL-2. HYAL-2 is on the outside of your cell. Let's just I'm not going to draw the real shape right now, but it's on the outside of the cell. And what it does is that when it encounters HA, it cleaves it. So we like this. We know that HYAL-2 is expressed globally. Which means it's expressed in all different cell types of the body. However, we're going to focus on one in particular, platelets. Okay, but let's remind ourselves what platelets are real quick. So platelets are this incredibly small cells, in fact they're sometimes called cell fragments, that when you have injury, you will actually start to see them form a clot. So they stick to one another. Look at that. It's forming a clot. So this is one platelet over here. And this is a clot or thrombus, sorry. Thrombi is multiple. Okay. However, on the surface of the cell, let's zoom in real quick. On the surface of the cell is also HYAL-2, which will allow it to cleave HA. Why do we care? Well, in IBD, platelets, let's say that they become effectively dysfunctional. Now, that doesn't mean that they can't do their job necessarily. They still clot, but they're hyperactive. And they don't perform functions the way they should. What I forgot to add on my whiteboard here is that if the platelets in IBD don't function properly, their HA metabolism via their HYAL-2 enzyme may be disrupted. This may be driving the inappropriate increase in the HA cables we were discussing earlier. And so this paper ultimately is asking, how does platelet HYAL-2 connect to loss or reduction of that HA metabolism I was talking about, which by extension is platelet dysfunction in IBD. And now for the paper's key experiments as described by the exact same lady with a PhD. What follows next is a very brief description of each of the key experiments performed in this paper. I do re-describe them briefly during the figure readings, so do with that info as you will. Listen on, skip ahead, you do you. You control your YouTube video experience. You have the power. Okay, so here are some of the key experiments that we'll be going through. You'll also hear me call them assays. So we're not going to go through all of them, but these are the ones that will probably pop up the most throughout the paper. The first one is going to be Western blot. So this is how we measure proteins in a given sample. So what does that mean here? Well, what we do is that we have cells and we'll collect them in a tube. Doesn't really matter what size tube. And then we'll usually break them up with something called REPA. So this is our sample over here. And then what we do is that we're going to put them in a gel. So this is what a gel will look like. And so we're going to take our sample and we're going to fill it in each of these wells. And obviously you're going to have a different sample for each one. Right. Once we have that, we are going to let these samples run. So really quickly, let's just get these back out of here. So these samples are going to run with electricity. We're going to run a current through the gel, which will allow the samples to start separating on the gel. So you'll see that the samples here will start to move. Once you're done with a run, what you're going to do is then you're going to transfer onto a membrane. So what's going to happen is that we're going to take the membrane and we're going to allow transfer of these separated proteins from the gel onto the membrane. Amazing. Let's focus on this membrane here. Now, we have a membrane that has these proteins embedded in it, and we need a way to visualize them because while I have them shown here, we can't actually see them on the blot. The blot will just look blank to us. So in order to see them, we're going to label them with antibodies, usually fluorescing antibodies. And so we're going to let that incubate, and we'll be able to see our proteins. What happens is that we'll be able to see where they are due to a ladder, which I didn't draw earlier. But if we know the size of a protein, we know that our given protein might be on this lane right here. Let me choose a different color. We know that our given protein will be this lane right here. Some of the other proteins we will not look for. The other thing to note is that the antibodies that we're using to tag the proteins are specific to our given protein. So if we're zooming in, this is a specific protein, and the antibody will be specific to that protein. Anything else down here is probably junk. This is the part where I began to talk about flow cytometry. Don't ask me why. This technique is only used once in the paper. I just really want to talk about it, I guess. Our next experiment. Cell migration. You're going to see this towards the end of the paper. And it's quite simple, really. We have a well that will have media in it. And then we have another well right here that will be dipped into the media. And it's important to note that these will have pores in them so that things can go in and out of them freely. And then what we do is that we coat with cells. These are cells right here. What happens in this assay is that we will add maybe some more cells, like a T cell right here. And then we will spike the media with, chemicals. It depends on the assay. So you might have TNF alpha, you might have even bacteria. It depends. What we're going to do is we're going to look for the migration of this cell from here through the pores down to here. And that is what we are measuring in these assays. So now let's cover a more niche experiment that we're going to run in with this paper, which is the hyaluronidase assay. Okay, what this is that we have a metal atom right here that will be coated with hyaluronan. So you have hyaluronan here. It's just like a little glycocalyx coat here. What happens is that when we have hyaluronidase activity, we're going to be cutting these HA, and what it'll look like is this. These are cleaved HA, and they're going to be sticking out. Then what we do, we will allow them to phosphoresce, which means glow. So these are now all glowy, glowy. Glowy, 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 glowy. We then measure that phosphorylation. And so if we have hyaluronidase activity, we will have data up here. If we don't, we will have data that's down on this end. No activity. One last thing, there's going to be a common experiment that we're going to see throughout the paper. It's called DSS colitis. And it's a mouse model assay. So we usually have mice in the lab. And what we're going to do, we're going to give them colitis. And what we do, this is their water supply. And we're also going to put a molecule called DSS in there, and they're going to drink that. Over the course of 7 days, so 1, 2, 3, 4, 5, 6, 7, this is days, we will monitor them because they will get progressively sicker. And then at the end of the 7 days is when we collect all of our samples from them. And what we're measuring is we're going to be measuring weight, fur condition, because mice are known to groom themselves, and the less they groom, the sicker they are. We'll also look at mobility of the mice, and then we'll also look at the colon itself, like the colon length. And then we'll also look at their stool, if there's blood in it. And this will all make up their score, their colitis score. The higher the score, the worse it is. I forgot to describe a key experiment called an ELISA assay. Due to Adobe hating me during this editing process, I'm going to take the L and not make animation for this one. What you need to know is that it's where we use glowing antibodies to measure the amount of a protein in a sample. And now the paper figures, as described by the PhD lady who is also learning how to record on her iPad. Okay. So we're going to walk through the paper much like you would for a class or for a journal discussion. So this here is figure 1. And what we're going to see here for 1A, this is a cut of a mouse colon. So really quick, what you're looking at is if we're looking at the colon, like we're looking at it down this way, and we have we've cut it this way. And so we're looking straight down the colon. And what we're going to see is we have our wild type mice on the left and our knockout mice on the right. Remember, we have completely deleted the HYAL-2 gene in all of this mouse. So it's knocked out in every single cell. That will be key when we get to later experiments. But this is an effect of what does it look like just at base, let's call it. So here is when you have the mice healthy. They are not sick. So their colons look relatively the same. They have the same shape. They have the same crypt structures. So what you're going to be comparing it to is wild type to knockout. However, when we put them on that 7-day DSS colitis model, which is a closed laboratory replica of colitis, we actually can see a major difference. So even here we can see that there's some swelling in the submucosal layers right here, even for the wild type one. Like the structure of the colon is a little bit sicker. But then when we look at knockout, it's even worse. Like it's very swollen. And that's where we have zoom in over here. That's why we have this whole section. In the labels, we have the lumen, which is just the space in between. Then we have the submucosa, which is this space right in here. And then we have this muscularis mucosa layer, which is this right in here. I'm just going to erase that for ease after I pointed that out. And what we can see here is that for 7 days, we still have swelling in our wild type. Like this is, we are swelling here. Like that's quite a lot of tissue. If you look at the tissue that's on right near the lumen, it's not looking fantastic. It's, I would say maybe okay, but it's not a very defined structure compared to it would be when it was healthy. And if we zoom in, we'll see these white dots occasionally, or this shape right here. That is what we call angiogenesis, or the formation of new microvessels. And the sicker you are when you have colitis, the more those will appear. So when we're looking at how sick a mouse colon is, we're looking at all those structures. So that's the wild type. But when we look at the knockout, it's even worse. Like, look at how sick this structure is right here. And I mean sick as in diseased. And then look at the swelling. Like, look at how big the submucosal is. And then remember those crypt shapes that we were showing earlier? Like, they're practically gone here. And then we can see a lot more of these shapes, which is, those are the, again, vascular structure. The sicker you are, the more you're going to have of those. And so, when we're determining how sick a mouse is, or even a patient, that's what we're looking at. And we'll be scoring those in a little bit. Now to go on to B. B is where you're looking at the weight change. And so whenever we put our mice on DSS colitis model, they will be monitored throughout the 7 days, and they will start to lose weight. And so what we're seeing here is that, yes, we have our weight loss, as you would expect on the wild type, but for the knockout has even more drastic weight change. Look at that. An average of about negative 10% change. They're losing about 10% over the course of the DSS colitis model, and that's significant for a mouse. And now we go to the disease activity index here. The disease activity index is what we're using to determine how sick a mouse is based on a couple of different parameters, like how is it maintaining its coat? How is it moving? Is there blood in the stool? And then we also look at how much the mouse's colon has changed. And so the higher that number, the sicker the mouse is. And so as you would expect for our no diseased mice, nothing has changed between them. They are not sick. But when they are sick, look at this. So our knockout is significantly sicker compared to our wild type after 7 days of DSS. And then we go to the histological score, which is what we do when we're scoring these guys. So again, what I was talking about before, the more like structural damage there is, the higher the score will be. And to no one's surprise, yes, the wild type right here have a high histological score. They are still sick after 7 days. But the knockout, again, has a significantly higher disease histological score. They are much sicker. And then we look at the colon lengths. This is where we're actually measuring them. When you put mice on DSS colitis, they will slowly begin to have their colon shrink just from the inflammation. And again, lo and behold, we have a worse outcome for our knockout mice after 7 days of DSS. So even just here, we can see that when we knock out HYAL-2 just everywhere on our mice, it's a worse outcome for DSS. Next, we're going to start zooming in on platelets. So what we did here is something called a Western blot. It's where we take our cells. These are cells. And then we grind them up or lysate them. And then we take that lysate, and again, we run that on a gel, stain the gel, and then we can see the proteins there that will look like bands. And that's what we see here. So you're probably wondering, what the heck is this and what the heck is this? So here, HYAL-2 is the protein that we're interested in, but this beta-actin is a control for us. What it is that for every single Western blot you do, you try to choose a control protein that shouldn't change even under disease. And what this helps us look at is maybe there is a difference between just how much you were able to collect between samples. You know, you might have one sample over here that just has a ton of protein. And then you might have one that just has a little bit, just because of, that's life. Sometimes that's how you sample. And so what that will look like on a gel is that you will have a very dark band for the protein you're looking for, and then you'll have a light band that you're looking for. That might tell you that there's a difference between them, except when you look at the control bands, you'll notice that you're like, we just didn't have enough protein in that other one. That will change how you analyze it. Anyway, so that's why we have that set up the way we do. Continuing on, what we see here is that we have our healthy mice. Over here, these are wild type mice. And we can see the presence of our HYAL-2 bands. And then we can see, when we put our mice on DSS, that the bands become lighter. Look at that. And that is quantified on this graph over here. Again, you're normalizing it against the beta-actin. That just means you're taking the value of what you found for your protein, and then you divide it by the value of control. And so that lets you make sure that that you are taking into account the differences in how much protein you collected between samples. So sweet. DSS results in platelets. And remember, these are platelet lysates. I forgot to mention that before. So platelets lose their HYAL-2. And then over here, there is a hyaluronidase activity. This is an assay that has gold in the center, a gold atom. And on the outside, it actually has HA that's around here. And then when we have hyaluronidases that are able to clip, what that will result in is the release of those HA, and then that will make everything glow. So that's how they're measuring hyaluronidase activity. It's a really clever assay. And we can appreciate, with the decrease in the HA, in the hyaluronidase that we saw up above, Of course, we see a decrease in that hyaluronidase activity. That makes sense. We just show that there's a decrease of the protein, therefore there's a decrease of the activity. And now, here we are looking at platelet count. How we look at that is through something called a hemovet. It just counts your platelets. And then we have serum over here. We are measuring how much HA has been released into the serum. Okay, so let's take this into account. We have, as expected, a decrease of platelets in our sick knockout platelets, and especially compared to our wild type. But this actually follows the trend that we have over here, where our knockouts just have less platelets than the than the wild type. Wow. So what we have here, less platelets, even knockout, no matter what. But what's exciting is that when we measure the HA that's floating around the serum, again, thanks to the hyaluronidase activity, we had HA like this, you cleave it, therefore there's going to be more floating around in the system. And even with less platelets, our sick knockout platelets are able to still have produce a higher amount of HA in the serum. Or at least that's what we're suggesting with this. There's a couple more things we need to do to prove that that's actually the case, but this is pretty interesting evidence. So, moving on to figure 3. This is again another colon slice from our mice. However, this is a bone marrow experiment. What does that mean? Well, we take our mice and then we swap out, this is a weird thing to say, but that's essentially what's happening. We swap out the bone marrow. You don't do the bones, that'd be too weird, but you can actually swap out the bone marrow to where I can put bone marrow from my knockout into a wild type mouse so that it's only the bone marrow that has the knockout, the hyaluronidase knocked out. Because you're doing a surgery and because that is inherently inflammatory, cutting into something and then injecting something into you is inherently inflammatory. You do the same thing as a control. You're going to take a wild type bone marrow and put it in a wild type bone marrow. Fantastic. So what we're going to have here is what we have, again, after 7 days of DSS colitis. As we expect, zooming in over here, we still have the swelling, the swelling here, we still have some of the crypts being lost up here. It is looking sick. But when we look again at our transfer, which is where we have a wild type mouse, and just the bone marrow of the mouse, of the knockout mouse, we see it looks sicker. It's more swollen. We've lost the crypt structure. I don't think you can see very well, but we have more blood vessel formation. And so why is this important? Well, bone marrow is where your immune cells come from. So what's great about this experiment is that you're able to get, knockout platelets, leukocytes. What's great is that these cells have the gene knocked out, and it's only those cells. The rest of the mouse is, wild type. Continuing on, when we have donor wild type into wild type, we still again have some weight loss. But when we do knockout into wild type, again, we have this drop in our weights, much more so than what we see in our wild type. So the immune cells are driving this change. And again, when we look at the disease activity index, it's worse. Histological score, it's worse. And then colon length, actually the colon length didn't really change. Did not change. Which, it's still exciting. All the other aspects were showing that the immune cells were driving those worsening symptoms. So now we've done the bone marrow experiment, but what's the next experiment to do? Well, these researchers decided to now do a rescue experiment. So what this is a rescue. And what they're doing is that they're taking the mice. This is knockout mice. Now we're no longer dealing within wild type mice. This is the knockout mice. And remember that the knockout mice generally have worsened symptoms, but we're going to actually add wild type platelets and put them into our knockout. And again, just for control, you're going to have knockout into knockout. Now what you're trying to do is that you're trying to see if what we're seeing is primarily platelet driven. It will, or you hope it will, improve the disease. And so what we have here is, again, another 7-day DSS colitis, and we have that characteristic sick colon look in our knockout. That's what we saw earlier before, but when we add in wild type platelets, we actually see a rescue of this phenotype. Look at this. So we have, again, the swollen submucosa, we have the loss of the crypt structure, with those angiogenesis right there, and, look at how sick that exterior of the lumen barrier is. But when we put in the wild type platelets, even after 7 days, look at how healthy this looks. Look at there's less swelling. There's still some good crypt shape. Not as much angiogenesis. This looks much healthier. It's still sick because you put it on 7 days of DSS colitis, but it is so much healthier. All right. So here, what are we doing here? Well, here we're showing that we actually have, when we put in wild type into knockout, we still have a HYAL-2 exposed on our platelets up here. This is a flow cytometry, and this is just telling me the platelets that you have put in there are still successfully going around in your mouse. So this is primarily telling us, hey, we have what we did, the transfer was successful. Huzzah. And that's supposed to be a mouse right there. Forgive me. All right, let's look at the weight change again. So with weight change, this is interesting. I actually would have flipped these just because that's what they were doing up above. Because what's important is that the wild type platelets into the knockout improve weight change, it improves index activity, a histological score was healthier, and we actually do see a change in colon length. So that's exciting. So remember, just changing out the bone marrow didn't really do much for our colons, but changing out the platelets did something. So what we see here is that it looks like platelets are driving difference in symptoms. That's what we can take around here. And again, that's what the figure 4's title is, but. All right, what's next on the list here? Aha! So now we're doing some ELISA assays, and we're looking at what is floating around in the blood. So here we have TNF-alpha and IL-6. So the reason why we're looking at TNF-alpha here is that TNF-alpha is known to drive formation of heavy chain HA. And if you remember what heavy chain HA is, remember that we have our, HA that's usually on the outside of the cells, and then heavy chain HA is what is used as landing platforms for your infiltrating cells. It's what's used to allow platelets to land in the area, etc. So TNF-alpha helps drive this change. IL-6 is another cytokine that is generally known to be pro-inflammatory, and also, they didn't mention it in this paper, but I do know that it is associated with hyaluronidase production and activity. So, let's look at the TNF-alpha. So, when we have no platelets swapped, but they're healthy, as expected, we don't have a pro-inflammatory cytokine. That's expected. Now let's look at when we have our wild type versus knockout and we haven't swapped out the platelets. As expected, actually, we have an increase in TNF alpha in our knockout, which is important because that is, as we saw before, knockout has worse symptoms compared to the wild type. So, worsening. However, when we do our, and again, I would have swapped these just for ease of read, but what we have here is our knockout in our wild type platelets into knockout and knockout platelets into knockout. And this actually improves like how much TNF alpha is in your blood. That means it has decreased. And so this, I say improved because you will have less pro-inflammatory signaling right here, and this one has more. Right, so now let's go on to the next one. And again, we see the same trend. When they're not sick, no TNF alpha. And then when they are sick, we do have an increase, but more so in our knockout. And then we have this rescue experiment here where we actually show that we have a decrease in the knockout here. So if you really want to have it down and dirty, like what are we really looking at? It's this comparison right here showing that our addition of wild type platelets, while it doesn't necessarily rescue it completely, this definitely lowers the inflammatory cytokines for both. All right. This is more cytokines, and again, this is...
And it was at this exact moment that my brain decided to have a brain fart, and I completely forgot how to read a graph. So after I collected myself, I restarted on C.
All right, picking up where we last left off. So now we're getting into C, D, and E. This is going to look very similar across all of these graphs. What we need to know is that F480 positive is how we're going to measure macrophages. Ly6 positive is how we are going to measure lymphocytes. And MPO, this is how we are measuring neutrophil activity. Okay, so that is what each of these are looking at. 3 different immune cells and different ways of looking at them. So what we can look at is the first 2 here is that this is again with no change in platelets here. And so obviously when we have DSS colitis, the knockout has more immune infiltrate or immune cells here. And that makes sense. The higher your immune cells, the sicker you are. And that's been consistent across all of these findings. And again, that's true if we look at the platelets again over here. DSS colitis, we have higher lymphocytes in the area. And then as well, if we compare these 2 here, the neutrophil activity is also higher. Now what we want to know is where we have knockout, where we have replaced it with either wild type or put back in knockout platelets. And what we can see here is that for our DSS colitis model, there is a reduction in all of these when you add the wild type platelets back into the model. That's really exciting because again, this is showing that when we add the cells back in, we can rescue a phenotype. And what you can even look at here, I'm going to choose a different color, is that this becomes more similar to this. So again, this is us rescuing the phenotype here. Now, it's not exactly the same, but we don't have a show of significance between the 2. And so that's why there's no stars up above, etc., etc. Okay. Let's head down to figure 6.
Okay, so figure 6 is some more staining here. And so these are at 2 different objectives. So this is close up, far away. Okay, and again, this is all in the DSS colitis model. We gotta just continue remembering that. What we are looking at here is green stain is going to be equal to our hyaluronan. And then we have red. That's going to be equal to inner I alpha I. I alpha I is what we find in our heavy chain hyaluronan form, which is the pro-inflammatory form of this. So what we should see is if it's more inflamed, it's going to be more red. And then we have in blue is the nuclei. Which is another way of trying to look at immune cells. Okay, these are healthy. This is what you should see. And we can appreciate there's not a lot of green, not a lot of red, and, some blue. I know I'm writing over the images, but you can see that here. Not a lot of green, really no red. However, once we get into the DSS over here, so what we can appreciate is that we can start seeing red, we see more blue, and we also see way more green starting to come up. It's obviously worst over here in the knockout. Look at this. So you have all these blue here, and blue is going to be equal to more immune infiltrate. Okay, what does that mean? It means that the immune system is hyperactive. It's on alert, and so it's sending more cells there. And then we can also see that that there's definitely more green. Okay, so just from here, we can tell that the knockout not only has more green, but it also has more red, which equals more pro-inflammatory healthy HCHA. Okay, but let's go here because this is where we're attempting to see if we can actually rescue. So when we add our knockout, our wild type platelets back into our knockout, we now see that this looks more like this right over here. I will erase that just for ease. But there is a rescue phenotype. So we have lower green and we have less red, and we definitely have fewer blue. Now we have over here, when we just add knockout into knockout, this looks very similar to this. Okay, so this is showing if it's more inflamed, it's going to be more what is happening for like hyaluronan. Because remember, ultimately we're looking at hyaluronidase activity, and really we want to figure out what is happening to the pro-inflammatory form of HA when we have a change in platelet hyaluronan during colitis. So this is where we're starting to get more into a model system. Or we were already before, but now we're really looking at HA. Okay, continuing on. So now we're looking here, we're actually measuring the tissue HA, which is what we were looking at up here, but now we're actually measuring it, and we can appreciate that even baseline, we have more HA in the tissue here. And then we definitely do when we have this knockout on DSS colitis. So look at that. But we actually show here, this is what's fascinating, is that there's no real difference between these two in terms of the amount of tissue, the HA that's in the tissue. However, when we look at the pro-inflammatory form, we can actually appreciate that the wild-type platelets into the knockout actually decreases the pro-inflammatory form, of HA. So that's what this is showing here. And then lastly, what we're looking at here is something called vascular permeability. And what you need to know is that is the leakage of your tissue. And if you're really sick, the higher your leakage will go, which is what we see here in our knockout compared to wild type. This is sicker because there's more leakage. Sorry if you can't read that. However, when we have this knockout, wild type platelets into knockout here, again, we have this rescue where our platelets make our mice less sick. What this is showing us is that the platelets are the ones that's driving the difference between some of the phenotypes or some of the symptoms that we're seeing in our disease or colitis disease. Specifically, and what this figure shows really, is that this pro-inflammatory HA metabolism or formation is directly associated with the platelets in your system and whether or not they have that enzyme. Now we're getting into people. We've done everything in mice previously, but now this is the people's time to shine. So that's when we're going to see little microvessels from patients. And then down here we have our mice. So what we're looking at here is a microvessel, tiny little vessels that's pulling blood throughout your system. And what you have is a cut of the tube, much like how we had earlier, where if your microvessel looks like this, we're cutting it right here and then looking straight down at it. This is an eye. And so we're looking straight at that. That's what we're seeing here. But you'll see that when we stain for red, which is again just our pro-inflammatory form of HA, we don't really see red here, but we definitely see it in our IBD patient here. We also see more green on the outside. We also see, I don't think we can really appreciate it here, of the increase in immune cells, which again is in blue. Just to remind us, green equals hyaluronin, red is equal to the pro-inflammatory form, or the HCHA, and then blue is equal to immune cells. And you'll note that this looks very similar to our models that we've been using. So these are our wild type DSS. I will note that I know I drew that there. This should also look very similar to this, as these are all sick. This is the only healthy one up here. Okay. Now we're going to be looking specifically at hyaluronidase activity. So the hyaluronidase activity, nope. 1, 2, 3. So what we're looking at here is the expression of HYAL-2 on our platelets. And as expected, let me draw this, we have a reduction in our IBD patient platelets, which is shown over here. Again, we have, So what we have here are cells that have been isolated from platelets, and these are called human intestinal, microvascular, endothelial cells, or HYMIC. As the name suggests, they are the cells that make up those little microvessels we saw earlier, or essentially. I'm oversimplifying a little bit, but what you need to know is that we are looking at the expression of HYAL-2 in these cells in patients. So this is our IBD patients versus our healthy patients. And we can just see immediately that our IBD patients have a reduction of our proteins in general. What we did next was we wanted to know whether or not TNF alpha regulates HYAL-2. The reason being is because TNF alpha, which is what we're dosing right here, is known to increase HA synthesis. And so what happens though when we add that into the system? And so what we did was that we did some cell culturing. So what we have here is no TNF alpha and then addition of TNF alpha. No TNF alpha, TNF alpha. And again, if you're wondering what is this gap DH, that is just your standard. That's just what you're comparing everything to. And then what we have over here is our measuring of HYAL-2. And we can see on this Western blot, we have a reduction of the blot here. That's really cool. Okay. So what we've shown here is that when we expose TNF alpha to patient cells, we can reduce the TNF alpha on there. Now let's move on to E. This is a cell migration assay. So what we're doing here is that we're looking at how well cells can pass across a cell barrier. And what's happening here is that we're adding a lot of things, but don't worry, we'll go through we'll go through each one. And so one set we're adding TNF alpha, or we're not. Another set we are adding platelets, or we're not. Here we have addition of platelets, and then take out platelets. Next, we have an antibody that blocks HYAL-2. We've only done the one of that. Then we also did hyaluronidase, which will cut up the HA. Okay, so how are we walking through this? Well, this is a standard cell migration if you just let the cells sit there. Sweet. That is under normal conditions. The next one is when we add TNF alpha. And TNF alpha will increase HA, or the HCHA, which will then allow for more recruitment of your cells. So that's why you see this increase here. Amazing. Now let's move on to the next one, which is where we add the platelets only. And the addition of platelets really doesn't do much. However, when I add TNF alpha, I also see that increase here. But note that this, and forgive my drawing here, this is still lower than the cell migration when we just add TNF alpha. So the presence of platelets does do something. So the presence of platelets, it does increase it just a little bit, but not to the point where if it's just TNF alpha. Okay, now that may baffle you for just a second until we get to the next one. Let me fix that real quick. When we get to this one, this is where the money making is happening. So what we were adding here is TNF alpha, platelets, but we're blocking the platelet hyaluronidase activity using this anti-HYAL-2 antibody. So what this is doing is that we are blocking the platelet activity, and so it looks more like we just added TNF-alpha. These look very similar to each other. So this here is suggesting that, hey, platelet HYAL-2 is a major component of cell migration. And again, it's important that we say platelet HYAL-2. Okay, and last but not least, then we have the addition of hyaluronidase. This is to remind you that TNF-alpha's primary role in the cell migration is HA increase, and so if we get rid of the HA with the hyaluronidase here, then we have a drastic decrease. So this is a control, but it's still important to show. I know that one was quite a bit of a doozy, but we're almost there. Let me erase this just for simplicity's sake. Okay. Next, what we're looking at is how much HA is circulating in your body. That's what this next one is about. All right, and we're going to go through each of them. When we add TNF-alpha, we have a little bit of increase of HA. A little bit. That's just for simplicity's sake. When we just add platelets, not much is happening. But when we're adding them together, we can again see that TNF-alpha will increase the amount of HA, or HCHA specifically. And then, when you have more of that, well, you're going to have more fragments from the platelet hyaluronidase activity. So this makes sense, and it's important to note that these fragments are pro-inflammatory. That means that these fragments can be released into the system to just create more and more of a reaction. Your body can get sicker and sicker and sicker. All right, and then last but not least, just to show that this really is being driven by hyaluronidase, then we add a blocking antibody. It's not quite as drastic as you would like, but it's still very significant. So this is, again, another control. All right. Last but not least is G. This here, let me just double check this. This is a lot. Okay, so G is where we have, again, those Hymex from the patient platelets over here. And what we've done is that we've taken them again from healthy people and super sick people. And how this is going to tie a lot of things together is say, hey, you know how we've been noticing that, when you're super sick, you have an increase in TNF-alpha, which will then increase the cell migration, and then your hyaluronidase on your platelets, which we have over here, should help control some of that migration. And remember, our IBD patients have a reduction of HYAL-2 for platelets. What is this showing? Why do we care if it has, if we have decrease in HYAL-2? Well, because we have a decrease in HYAL-2, our IBD patient platelets are going to have a ton of TNF-alpha. Because TNF-alpha is released when you're super sick. And if you have TNF-alpha, again, look at E. I'm going to draw this over here. If we look at E, we know that TNF-alpha will increase the amount of cell migration across these Hymex. And we also know that when we have that, our healthy patient platelets will still have the hyaluronidase, HYAL-2, to keep that under control, but then we lose that hyaluronidase, which will then lead to inappropriate recruitment and migration across the Hymex. So this one, as simple as it is, ties it all together. Phew, that was a lot. All right, now time to wrap this up. So ultimately, why do we care? Why is this paper such a big deal? Why was it published? And there are many reasons why this paper is important. I'm going to just highlight 2 of them that were pertinent to what I was studying during my PhD. So, first and foremost, this paper highlighted a new area of study for possible IBD treatments, which will specifically look at platelets. A lot of times people look at the other inflammatory cells to try to regulate IBD, but this paper started to highlight how focusing on platelets could also help alleviate some of those symptoms. And then the other big point of this paper is that it was the first to show that platelets regulate those white blood cell recruitments to the sites of inflammation during IBD via the platelet HA degradation activity. So there is a direct link between there. Again, very exciting. So at the end of every paper, they're going to have areas that still need to be studied or questions that still need to be answered based on the data that was provided. And for this one, this paper really highlighted that the research that he did in this paper was looking more at how platelets protect during the initial stages of IBD. However, we know that platelets, as we said earlier, are dysfunctional. And so how does this loss of hyaluronidase activity then lead to platelets making IBD worse? Because remember, if it's protective, well, at what point does it no longer become protective? And at what point does this add to the inflammation during IBD? You know, just for my paper. That's all.